DNA sequencing begins with a double helix. Within a double helix there are two strands. Each strand contains molecules called nucleotides. There are 4 nucleotides that exist in nature. These are adenine, thymine, guanine, and cytosine. The bonding rules for nucleotides are that adenine always bonds with thymine and guanine always bonds with cytosine. To sequence DNA, we must obtain one single strand. This is done by splitting the double helix of DNA using restriction enzymes. This becomes our primer, the list of nucleotides or code that we wish to sequence. We set up 4 reaction mixtures. In each reaction mixture, we add a primer. This is our single strand of DNA. We also add DNA polymerase. This is an enzyme used to attach free nucleotides to our primer. We add DNTPs, also known as free nucleotides. These will help complete our strands. And finally, we add DDNTPs. These are our chain terminating nucleotides, each one marked with its own fluorescent color. Adenine is marked with red, thymine is marked with green, guanine is marked with brown, and cytosine is marked with blue. The results we obtain in the end are that each strand has been completed to a specific nucleotide, which in turn has been marked by a fluorescent color. In the end, we end up with every single nucleotide fluorescently marked, but all on different strands. We then use electrophoresis to put our strands in order from largest to smallest. This is done using agarose gel and a positive charge. The larger strands get stuck in the polysaccharide chains in the gel, and the smaller strands squeeze through. They reach the bottom quicker. DNA has a slightly negative charge and is pulled through the gel towards the positive charge. Here is an example of a 5 nucleotide long chain, with each nucleotide attached to a different primer, but fluorescently marked, identifying its nucleotide. When all the results are added together from this experiment, we can see the original DNA sequence. It is AGACT. When using an automated machine, the machine lines up your strands from largest to smallest, and then it can post the results on a sheet as shown here at the end. This is very useful, as then all we need to do is take a look at the final letter of each strand, and we can add them all together to obtain our original code.